This video demystifies CRISPR gene editing, where it came from, how Cas proteins and guide RNAs cut DNA, real-world uses from medicine to agriculture, and the technical, ethical, and delivery challenges shaping its future. Chapter 1: Foundations. DNA, the genome, and CRISPR's origins. CRISPR stands for clustered, regularly interspaced short palindromic repeats, a bacterial adaptive immune system discovered from sequence patterns in the 1980s and linked to antiviral defense in the 2000s. Bacteria store short spacer sequences, typically a few dozen base pairs, copied from invading phages. These spacers guide Cas nucleases to matching viral DNA. In humans, the genome contains roughly 3 billion base pairs. Manipulating even a single base can change protein function. The commonly used Cas9 enzyme was derived from Streptococcus pyogenes. Understanding this cellular memory system explains why we can program CRISPR. Bacteria already evolved a sequence-based targeting mechanism we can repurpose for precise editing. Chapter 2: Mechanism. How CRISPR-Cas9 locates and edits DNA. At its core, CRISPR editing uses two components: a guide RNA, about 20 nucleotides complementary to the target, and a Cas nuclease, Cas9. Cas9 first recognizes a short adjacent motif. Cas9 prefers the pan sequence NGG. Then the guide RNA hybridizes to the target, and Cas9 makes a double-strand break. The cell repairs that break by non-homologous end joining, NHEJ, which is error-prone and often creates small insertions or deletions to knock out genes, or by homology-directed repair, HDR, which can copy from a supplied DNA template for precise edits. HDR efficiencies vary widely and are typically low in non-dividing cells, while NHEJ is active in most cell types. Chapter 3: Applications and concrete examples. CRISPR has been deployed across research, medicine, agriculture, and diagnostics. In medicine, ex vivo editing of hematopoietic stem cells is in clinical trials for sickle cell disease and beta thalassemia, with initial patients achieving therapeutic increases in fetal hemoglobin. In agriculture, a 2016 CRISPR-edited white button mushroom disabling a browning gene demonstrated faster-to-market improvement compared with transgenic approaches. Gene drives in laboratory Anopheles mosquitoes have achieved population suppression in caged trials, offering a malaria control concept. Diagnostic tools like Sherlock and Detector use Cas13/Cas12 to detect viral RNA or DNA rapidly. Each example highlights how edit type and delivery method determine feasibility and timeline. Chapter 4: Challenges, Safety, and the Next Wave of Tools. Key hurdles include off-target edits, delivery into specific cell types, immune responses, and ethical limits on germline modification. Delivery vectors matter. AAV vectors have X1 4.7 kb capacity, constraining payloads for Cas9, approximately 4.2 kb, and promoters, smaller nucleuses, example, Cas9, approximately 3.2 kb. Or split systems are used. Newer technologies reduce risks. Base editors chemically convert single bases without double-strand breaks, e.g., CT or AG. And prime editing, 2019, can install small insertions, deletions, and all base substitutions with fewer byproducts. Regulatory frameworks and societal debate will shape which applications advance to clinical or environmental use. CRISPR repurposes a bacterial immune strategy into a programmable molecular tool. Understanding guide design, PAM requirements, repair pathways, NHEJ versus HDR, delivery constraints, and newer approaches like base and prime editors clarifies both CRISPR's power and the technical and ethical limitations in guiding its responsible use.